Hello, microbiome fans. This is Tracy, and I wanted to talk to you specifically about how ZenBiome, our new Microbiome Labs product, specifically addresses stress in regard to the hypothalamus pituitary axis and the hypothalamus pituitary thyroid axis. So let's just, as a quick review, keeping these terms in mind, we've got the hypothalamus releasing the cortisol releasing hormone to the pituitary. And this, of course, is going to be the primary feedback control of how the adrenals producing cortisol have an effect in this loop with the brain. So as you produce enough cortisol, it then will inhibit this cycle. In the thyroid, very similar thing. You've got the hypothalamus releasing thyroid releasing hormone to the thyroid, which is going to produce T4, which is going to convert to T3, which is the feedback loop that stops that cycle. And then if long-term acute stress takes place, then it is going to inhibit the thyroid function. So how does ZenBiome help with this? Well, we know that what also inhibits these cycles is blood sugar and inflammation and stress in general. So we have launched this new product, and we're really excited that this has a primary effect on the inflammation as well as the feedback loop of stress involving cortisol in the HPA and HPT axis. So here are our preclinical studies showing the effect on inflammation. Our strain of Bifidobacterium longum 1714 induces interleukin-10 cytokines, which are anti-inflammatory in the spleen. The 1714 reduces the pro-inflammatory transcription factor NF-kappa B in mice injected with lipopolysaccharide in a mouse endotoxemia model. 1714 reduces pro-inflammatory cytokines IL-1 beta, IL-12P70, and TNF-alpha in the serum or spleens in mice injected with lipopolysaccharide in a mouse endotoxemia model. 1714 reduces the pro-inflammatory cytokines L1B, IL-12P70, and TNF-alpha in mouse spleens in a diet-induced obesity model. So the conclusion here is that the 1714 is an anti-inflammatory strain with potential to help with the inflammatory mediated stress. So this is really underlying the whole gut, brain, and stress connection as far as emotions are concerned. So here we see that the cognition of animals are affected, and here are the models underlying that effect. In the object recognition test, the Bifidobacterium longum 1714 fed mice discriminated between the two objects faster than all other groups. In the Barnes maze, the B. longum 1714 treated mice made fewer errors than other groups, suggesting a better learning. In the fear conditioning, B. longum 1714 treated group also showed better learning and memory, yet presenting the same extinction learning profile as controls. So this is important to see that it has not only an inflammatory effect, but also on the ability to learn and stress behavior. Here are the preclinical conclusions on behavior. 1714 and oxalytic and antidepressive effects in mice are linked to a reduction in the pro-inflammatory cytokine IL-1 beta. 1714 had a positive impact on cognition in terms of object recognition, better learning, and memory. So again, the conclusion is that 1714 is an anti-inflammatory strain with potential to help inflammation-mediated stress and cognition. So now let's look at the human studies. And here we see that the Bifidobacterium longum 1714 as a translational psychobiotic modulation of stress electrophysiological and neural cognition in healthy volunteers. You can see the study design here, and that the whole blood stimulation with TLR agonists. You can see the cortisol output and the reduction from visit 1, the placebo, and the reduction of the 1714 strain by the 1714 strain, and the overall stress response. So the human clinical trial conclusion is the 1714 reduced perceived stress and cortisol output in human male volunteer, and this was associated with a reduction in the pro-inflammatory tone of these subjects, demonstrated through a decrease in pro-inflammatory cytokines, IL-1 beta, IL-6, and TNF-alpha in the toll-like receptor stimulated whole blood from these subjects. Now, it was also shown in a clinical study that the 1714 alters brain activity compared to a placebo. You can see this is a very unique study where they are actually able to measure brain activity. You can see that they can see the actual brain scans and relate that to emotional response. So the clinical trial conclusion on brain activity strain was that 1714 reduced perceived stress and cortisol output in the healthy male volunteer, and this was associated with a reduction in the pro-inflammatory tone of these subjects, demonstrated through a decrease in pro-inflammatory cytokines, IL-1 beta, IL-6, TNF-alpha in the toll-like receptor stimulated whole blood from these subjects. 1714 activates the centers of the brain, correlates with improved vitality and control of the negative emotions. 1714 improves different indices of sleep in 2 separate studies. 1714 improves quality of sleep parameters such as improved social function and vitality. So we are very excited with the results of these tests on the launch of our new products, ZenBiome, and in combination with the gut conditioning of the spore products, and Megaspore is an amazing connection between the gut-brain, stress, behavior, and coping with stress and sleep. Thank you for your attention.